Vaccine scandal, Plasmidgate. What is it about DNA contamination in COVID-19 vaccines? Why is DNA a topic at all for products that contain mRNA as the active ingredient? mRNA is not assembled directly, but read from a DNA template. This is done in biotech production in bacterial cells. Bacteria contain small DNA rings, so-called plasmids. The DNA code for the COVID-19 spike protein is inserted into these plasmid rings using genetic engineering techniques. The plasmid is specifically customized, so to speak, for the production of the mRNA active ingredient. DNA itself cannot initiate the intended vaccination effect in the body, but can cause side effects. It must therefore be removed. For this purpose, the crude intermediate product is purified after biotech fermentation. In the course of such purification process, the small DNA fragments must be technically removed. However, analytical testing of vaccine batches by independent test laboratories in the US, Canada and Germany repeatedly indicate that residual DNA is contained in the COVID-19 end product for human use. According to these test results, the specified limit for DNA impurities in the end product is exceeded up to 350 times. The manufacturers remain silent. It is unknown if health authorities have initiated investigations. Is this a manufacturing and testing scandal? Were vaccine batches in Europe randomly tested for impurities by official control laboratories? Or only the manufacturer's test certificate, meaning paper-checked? For such new mRNA vaccines and such new mechanism of action, control tests would be expected, at the latest upon repeated reports on DNA contamination, which have been the subject matter of heated debate among experts since February 2023. It should also be questioned whether the method used by the manufacturer to test for DNA impurities, namely PCR, is the best method. With PCR, one must know what DNA fragments to test for. Only then one will get reliable and right test results. For the end product, the mRNA active ingredient is formulated as lipid nanoparticles to administer into humans. The same is to be expected for DNA impurities, which will then also enter human cells. How much bacterial plasmid DNA is actually contained in a COVID-19 vaccine dose? Is the set limit correct? What effects could DNA impurities have in human cells and to the genome? Who is liable? These and other questions must be urgently clarified. Until then, COVID-19 vaccination must be stopped immediately. GGI-Initiative.at, Austria.